Does that work?
Yeah, I can see that.
Yeah, perfect.
Okay, sorry, I just need to, I have a button I need to get rid of somehow, but I don't see it. Sorry, just give me a second. There we go. And then I'll go back and share. Okay, that should work, I think. Perfect. Okay. Hello, everyone. Thank you, Iker, for that really nice introduction, and thank you all for having me. It's really, really a pleasure. So like Iker said, I am currently a postdoc at SLU. I just started 2 weeks ago. So today I'm only going to be presenting some of my PhD work that I did at Uppsala University in the lab of Fabian Burki. And I'm going to be talking about 2 topics. So the first is long-read metabarcoding of eukaryotes and how we can do it. And then the second topic is how we can use these data to learn more about how eukaryotes have evolved. And here I'm going to focus on habitat transitions. So when I say eukaryotes in this presentation, I'm mainly talking about microbial eukaryotes, or protists. So the figure that you see on the screen shows the eukaryotic tree of life. And we often only think about plants, animals, and fungi. But these groups are just a few tiny branches on the eukaryote tree. And most diversity is, in fact, microbial. And it comes in all of these beautiful shapes, forms, and sizes. So just in case you're not so familiar with them, I'm going to show you just a few examples so you can bond with them a little bit. So we start with the diatoms. So these are photosynthetic algae in silica glass shells. It's estimated that there are 100,000 species of diatoms, and altogether they produce 20 to 30% of the Earth's oxygen. So that means every third breath of oxygen that you're taking in comes from this group of protists. We also have ciliates. Most of them are heterotrophic. There are a few parasitic examples, but here we see the ciliate Lacromeria, which is quite a ferocious predator, and it hunts with a sort of really long, extendable neck-like structure, which it uses to catch prey. There are also microbial vampires, like these vampirellids, and these suck out the cell contents of algae. And I'm going to show you one more example, and this is a really cute one. This is called Meteora. It has this really cute way of moving about. It almost looks like it's waving at you. So these are just a few examples of the vast diversity of microbial eukaryotes, which altogether are important primary producers, they're heterotrophs, they're parasites, and they're key players in global biogeochemical cycles. So how do we study this amazing diversity in the environments? The most straightforward way is short-read metabarcoding, or the most popular way currently. So this is where you take your environmental sample, could be soil, water, or air, and then amplify a short barcode of less than 500 base pairs. Most commonly, this is the V4 or the V9 hypervariable fragment of the eukaryotic 18S gene, or the SSU gene. And then this is sequenced with Illumina. Now this generates millions and millions of reads, and by comparing these reads to a reference database, it's a very powerful way of identifying things in your sample. And over the past decade or so, there have been many metabarcoding surveys that have been carried out, some on a truly global scale, like the Tara Oceans Project or the Malaspina expedition. There have been global soil surveys, and all of these different studies have generated large sequence data sets, either for the V4 or the V9 regions. And then altogether, they've allowed us to do things like study protists communities in different environments. And we've also discovered lots of novel diversity. However, this metabarcoding approach has one big limitation, and that is that these short reads contain very little phylogenetic signal. And that makes it challenging to answer ecological and evolutionary questions that rely on phylogenetic inferences. For instance, it's hard to know where a new lineage belongs in the tree of life when you can't build phylogenies. And so the first aim of my PhD was to overcome the limitations of the short Illumina reads by sequencing not just the whole 18S, but almost the entire ribosomal DNA operon. And then this would allow us to harness the greater phylogenetic signal of the long reads. So we started by doing this proof of concept study where we developed long read metabarcoding and how to analyze it. So for this study, we obtained 3 soil samples, and then we used general eukaryotic primers to amplify around 4,500 base pairs of the ribosomal DNA operon. This includes the 18S, ITS1, 5.8S, ITS2, and the 28S genes. So we then sequenced these long amplicons with PacBio on the SQL platform using circular consensus sequencing, which is also called CCS for short. So to overcome the error rate of PacBio sequencing, we developed this stringent curation pipeline for our environmental sequences. We filtered based on quality and we removed chimeras. And then at the end of this pipeline, we extracted the 18S and 28S genes. So we chose to not extract the ITS region because it's far too variable across large eukaryotic groups. And then we clustered our 18S and 28S sequences into operational taxonomic units, or OTUs, at 97% similarity based on the 18S gene. And then after discarding singletons, we ended up with 650 OTUs from our 3 soil samples. We then treated these 650 OTUs in a phylogenetic framework. And the first thing we wanted to do was to identify these sequences. What groups did they belong to? And for this step, we used only the 18S sequence because this gene has a much more comprehensive reference database or databases. So I'll show a very simplified version of the method that we used. This was a method we developed with members of the Stomatakis lab. And the way it works is that you start with sequences from a reference database. These are labeled sequences, and then you can build a tree with them, like so. We then used the labels at the tips of the tree to annotate the inner nodes. And then we could phylogenetically place each OTU. We can insert it one by one onto every single branch of the tree. And then for each placement, we can calculate the probability that the OTU belongs there. And this is represented by the size of this circle. So in this example, we can be very confident that this OTU sequence is a ciliate and probably belongs to ciliate genus A. So all of this information together is used to calculate the taxonomy of the OTU. And the result is this table, which lists the taxonomy for each OTU to the appropriate taxonomic level. It may not be down to species level, may only be down to family or order level, whatever is appropriate. And then for each taxonomic rank, we have an associated confidence level from 0 to 1. So this way, we're able to label the 18S part of the OTU. And then because it's physically linked to the 28S molecule, we can transfer the annotation to the 28S gene or the LSU gene as well. So now that we had labeled our OTUs, we used both the 18S and the 28S genes to construct phylogenies to investigate the diversity of our samples. And this resulted in this phylogeny, which spans all of eukaryotic diversity, or at least it covers all of the eukaryotic major groups. So in this tree, branches in orange are our 650 OTUs. And then the branches in black, these are reference sequences from databases for which we have both the 18S and the 28S sequence from the same species. So doing this allows us to visualize the diversity present in an environment in a much more meaningful way. And because this is a multi-gene tree, it has both the 18S and the 28S, we get a more robust phylogeny. We also observed that while some clades, like the metazoa or the animals, have lots and lots of references, you can see many black branches over there, other clades have very few, like the rhizaria. And this shows us that our databases are very biased towards well-studied groups. But also, more importantly, it shows us how powerful our method can be for improving our databases. Because with our taxonomically labeled environmental sequences, we can fill in these gaps in our databases, in particular for the 28S gene. So just to summarize this work, we showed that we can sequence environmental diversity with PacBio. We can do long-read metabarcoding, and we can analyze it in a phylogenetic framework, from assigning taxonomy to building more robust phylogenies. So now that we could build these dense environmental phylogenies, the next question was, well, what more can we learn about eukaryotic evolution? And here we decided to focus on habitat transitions, particularly between marine and non-marine habitats. So why habitat transitions? Well, they're very interesting events in general, right? Because adapting to a new, very different, physically different habitat, it's a major evolutionary event. For example, if you think about the move of vertebrates to land from the ocean. And these events can be very important drivers of evolution. One of the most difficult habitat boundaries to cross, and the one that I focused on, is the salt barrier. So this is the boundary between saline marine environments and the low salinity non-marine environments, such as soils and freshwater. And this salt barrier is very difficult to cross because adapting to this new salinity or adapting to this new osmotic pressure is very difficult. So we asked, well, how frequently have eukaryotes crossed the salt barrier? Are certain groups better at transitioning across environments? And in which habitat did major eukaryotic clades arise? To answer all of these questions, we generated a dense 18S, 28S eukaryotic dataset from several different environments. So we had some soil samples from forest soils to agricultural soils. We had some freshwater samples from lakes, also from permafrost thaw ponds. We had samples from the surface of the ocean and also some samples from the deep ocean, including the Mariana Trench. And then for all of these samples, we did the same thing as before. We amplified the ribosomal DNA operon, and we sequenced it with PacBio, this time on the Sequel II platform. And then after processing the data, we obtained more than 17,000 OTUs. And these were annotated the same way as before using our phylogenetic method. Okay, and so I'm going to show you the phylogeny that we recovered from these sequences. So this is a multi-gene tree. It's an 18S28S tree of annotated environmental sequences. So each of these colored little pie chunks that you see represents a major eukaryotic group. So in this tree, it's all environmental sequences. We did not include any reference sequences here. And with this phylogeny that we taxonomically constrained, we spanned all of the eukaryotic supergroups and the major lineages. And when we map on the habitat data, we can visualize habitat preferences across the global eukaryotic phylogeny. So in this outer ring, orange and yellow represent freshwater and soil habitats, and then the light blue and dark blue show marine habitats. And we can immediately see that soil and freshwater organisms seem to be quite closely related. And then we see the same case with organisms from the surface and the deep ocean. But we do see these really distinct clades of marine and non-marine organisms. And this indicates that the salt barrier is indeed difficult to cross, not just for plants and animals, but also for microbes. Now, while comprehensive, our tree, of course, does not cover all of eukaryotic diversity. We're missing many species. So we try to increase this covered diversity by incorporating available short-read metabarcoding data sets that were publicly available. Here we focused on the V4 region because it's slightly longer, has more of a phylogenetic signal than the much shorter V9. So some of the data sets we included are the Malaspina data set. We had neotropical forest soils, some global soil surveys, multiple freshwater surveys, etc. And this was a lot of work that was done by Charlie Beaver, who was a master's student in the lab at that time, and Daniel Volo. So in total, we obtained roughly 200 million reads, and we clustered them into OTUs, and then inferred phylogenies combining both the long read and the short read data. Here we used the PacBio phylogenies as backbone constraints into which we could insert these short read sequences. So the result is that we got these really comprehensive trees, and we were able to detect transitions that are missed by the PacBio data alone. And to illustrate this, I'm going to show you one example from the vampyrellid phylogeny. So this is just a small section of the phylogeny of vampyrellids, of environmental sequences. In the orange are sequences from freshwater and soils, and then in blue are the marine sequences. And in this particular case, all of these blue sequences came from the Illumina short read data. So now that we had these really comprehensive phylogenies, we can use ancestral state reconstruction to investigate different aspects of habitat transitions. And one of the questions we had was, are there certain groups that are more adept or better at crossing the salt barrier? And we can investigate this by looking at the number of transitions per unit of branch length. I'll show you with this cartoon over here. So the two colors represent, again, different habitats, the marine and non-marine. And in this phylogeny on the left, we see a single transition event from a marine to a non-marine habitat. In other words, infrequent transitions. While in this phylogeny on the right, we observe multiple or frequent transitions. So this way we could compare the phylogenies of different eukaryotic groups. And we saw that, yes, transition rates do vary very greatly across major eukaryotic lineages. So in this graph, the x-axis depicts so these are density curves, and the x-axis shows the global rate of habitat evolution or habitat switching. So it's increasing in this direction. And here we saw that fungi have by far the highest transition rates. And indeed, when we look at the fungal phylogenies, we see that the marine and non-marine lineages are very, very closely related. They have very tiny branch lengths in between them. So next, we asked, what were the ancestral habitats of eukaryotic supergroups or eukaryotic major lineages? And for this, we performed ancestral state reconstruction using base traits. Now, this figure is very busy, but I'll try to walk you through it. The figure depicts the eukaryotic tree, and then each branch or each tip represents a major group. For example, here we can see metazoa. So we have one branch representing the animals here. And then the pie charts at the base represent the likely ancestral habitat for that group. So again, we can look at the metazoa, and here we can see that our analyses show that metazoa likely first evolved in marine habitats, which is consistent with what we know. So we did this for all of the different groups, and what I'd like to point out, or what I'd like to focus on, is the two largest groups of eukaryotes. These are called the Amorphia and SAR. So each of these are much larger than the plant and animal kingdoms that we know. And what we see is that these two major lineages arose in completely different habitats. The SAR lineage, which includes groups like diatoms, ciliates, dinoflagellates, radiolarians, this group first arose in the Precambrian oceans. On the other hand, the ancestral Amorphian, which has now diversified into fungi, animals, choanoflagellates, amoebozoans, the ancestor of this group likely inhabited non-marine habitats. And then finally, we zoomed out even further to investigate where eukaryotes themselves first evolved. Now, this is tricky because we don't know the position of the root of the tree, but we tried the two most popular proposed positions, and that is Discoba, sister to all other eukaryotes, as well as Amorphia, sister to all other eukaryotes. And in both scenarios, it seems more likely that early eukaryotes inhabited non-saline habitats. And this was a very surprising result for us because we often tend to think of oceans as the cradle of life. But perhaps this result is not so strange because there is some evidence from previous studies of other great eukaryotic events occurring in low salinity habitats. For instance, the origin of plastids or chloroplasts. But on the other hand, we also need to take this result with a little bit of grain of salt, because in this study, we didn't sample marine sediments or anoxic environments or other extreme habitats out there. And these habitats may contain deep branching lineages that are missing from our trees. So it would be very interesting to see if we can confirm these results when we include even more data. So to conclude this study, we showed that the salt barrier has been a major factor in shaping eukaryotic diversity. We showed that transition rates vary greatly across eukaryotic groups. And this raises the very interesting question of why? Why is that? Why are some groups better at adapting to different salinities? We showed that SAR and Amorphia, the two largest eukaryotic groups, likely arose in different habitats. And we showed that metabarcoding, the combination of long read and short read metabarcoding, can and should be used to investigate evolutionary questions. And in general, I think we might be on the verge of this exciting new phase in metabarcoding, where we can look at protist diversity through an eco-evolutionary lens. And for this work, I'd like to thank a lot of people, Fabian Berkey, who was my supervisor, for all of his support, as well as all of the other members of the lab. We had some wonderful collaborators who provided us with these amazing samples. They provided data and developed methods with us. And just a quick note that if you're interested in any of the data that I presented today, you can find them online. So we've put up our taxonomically annotated long read sequences on Figshare. And if you're interested in short read metabarcodes, you can go to the MetaPR2 website, which has been developed by Daniel Velo. So this is data from 41 different data sets that have been processed in a standardized way with Data2. So you can check that out. And thank you for your attention. I'm happy to take any questions.